All right, our last screencast on chapter 12, cell division, we're going to look at the regulation of the cell cycle, how the cell cycle is regulated. Now, you guys have heard me talk a lot about how all of our metabolic processes, transcription, translation, protein synthesis, cellular respiration, photosynthesis, signal transduction, how they all need to be controlled. We need to be able to turn things on and turn things off. And all of these rules, of course, apply to the cell cycle as well. So the eukaryotic cell cycle is one of the most tightly regulated things that there is. There are so many hundreds of backup systems. There's hundreds of molecules that control the cell cycle. So it's controlled by a molecular control system, and it all controls whether or not a cell is going to divide. It determines whether or not a cell divides. How often a cell divides, the frequency of cell division varies with the type of cells. There are some cells that divide very often. Skin cells, sperm cells are one of those cells that divide most often, most frequently in the human body. Other cells divide hardly at all. Nerve cells divide once and then never divide again. So it really depends on the type of cell. Okay, and this is all controlled by molecules that control the cell cycle. So all cells are basically the same, but whether or not they're going to divide is determined by these molecules that control the cell cycle. Of course, our dear friends cancer cells completely escape the usual controls of the cell cycle, and they kind of divide whenever the heck they want. So when they first looked at trying to figure out what controlled whether or not a cell divided or not, they looked at signals in the cytoplasm. And the cell cycle appears to be driven by specific chemical signals that are present in the cytoplasm. And what they did for this, they did some experiments where mammalian cells at different phases of the cell cycle were fused to form a single cell with two nuclei. So they looked at two pairs of cells. One was a cell in S and a cell in G1. And what happened was they fused the cytoplasm of these two cells. And so basically one of the nuclei was in S phase, so replicating DNA. The other nucleus was in G1 phase, so just kind of hanging out and growing and doing whatever. Once they fused the cytoplasm, the G1 nucleus immediately went into S phase. Okay, so DNA replication began immediately. Same thing occurred when they took cells that were in M phase and cells that were in G1. And remember, cells in G1 have not replicated their DNA. They are not ready to divide at all. And when they fused the nuclei on these two cells, the nucleus that was in G1 immediately began to go through mitosis. So even though it hadn't replicated its DNA yet. Okay, so there we go. All right, so this kind of gives us evidence that there are certain signals present in the cytoplasm that controls what phase of the cell cycle the cell is in. Keeping the events of the cell cycle in the correct sequence, sequential events, is directed by a cell cycle control system, which is very similar to we call it a molecular clock. Okay, so everything has to happen at the right time. It's regulated by both internal signals and external signals. And the clock has these points we call checkpoints, where the cell cycle stops until the cell is given kind of the go-ahead, given the green light. And there are three main checkpoints in the cell cycle: G1 checkpoint, a G2 checkpoint, and an M checkpoint. And the G1 checkpoint appears to be the important one. If a cell gets a go-ahead to go past the G1 checkpoint, that cell is going to basically complete the cell cycle, go all the way around and divide. There are two other stops here, but basically it's going to eventually divide. So again, that G1 checkpoint, it's kind of, we call it the start point. Seems to be the most important. If the cell receives the green light, it's going to divide. It's going to complete G2, S, and M phases and divide. If the cell does not receive a green light at this checkpoint, it's going to exit the cell cycle and switch to a non-dividing state that we call G0. So if we get the green light, cell goes around, divides, completes the cell cycle again. If the cell does not receive a green light, it exits the cell cycle and enters a phase called G0, where it just sits there and doesn't divide. And G0 can last a couple of days, couple of years, or the entire life of an organism. Okay, so let's look at the two main types of molecules that are involved in controlling the cell cycle, and they are cyclins and cyclin-dependent kinases. Okay, so just a quick review here. We all know a kinase is an enzyme that phosphorylates other molecules. And we have these cyclin-dependent kinases, we call them CDKs for short. Okay, their activity is dependent on the molecule cyclin. Okay, so CDK activity fluctuates during the cell cycle because it is controlled by cyclin. And a cyclin is a separate protein, okay, that whose concentration varies within the cell cycle. That's why we call it cyclic. It has a high concentration during some parts of the cell cycle and a low concentration during other parts of the cell cycle. Okay. One cyclin CDK complex, and there are hundreds of cyclins and CDKs that all have a role in controlling the cell cycle, but one of them is called MPF, and that's the one we're going to focus on. That stands for maturation promotion factor. So this is a cyclin CDK complex that triggers a cell cycle past the G2 checkpoint into M phase. So let's just take a look at the MPF. So again, up here, we look at fluctuation of MPF activity due to cyclin concentration. Okay, so again, CDK, or cyclin-dependent kinase, is present at all times. Okay, but it's only active when it's bound to cyclin. Okay, so we look at the activity of cyclin, CYCLIN. We look at the presence of cyclin, and if you look here, okay, cyclin concentration is the purple line. As the concentration of the cyclin that binds to the CDK that creates MPF, that concentration increases. As it increases, it begins to bind to CDK and cause it to become an active kinase. That triggers the cell to go through G2 into M, and then the cell will divide. Once that transition occurs, we get rid of the cyclin, and then activity falls off. And then it keeps, so again, as we're coming up through G2 again, cyclin concentration increases. That causes a spike in the activity, pushes the cell through M phase. Once that transition is achieved, get rid of cyclin. That causes the CDK activity to decrease, and then the whole thing happens again through the next cell cycle. And if you look here, so as we're going into G2 phase, okay, cyclin begins to accumulate. Here is cyclin. If it's present, it will bind to CDK, which is present all the time, and form MPF. That causes the cell to go through the past the G1 checkpoint, the G2 checkpoint. Once that occurs, cell goes on its merry way. Cyclin is degraded, okay, and then CDK is no longer active. It's present, but not active through the remainder of the cell cycle. Okay. So again, and this is an internal signal that helps to control the cell cycle, one of many. Another example of an internal signal is if kinetochores are not attached to spindle microtubules, that sends a molecular signal that will delay anaphase. Okay. And then if we have external signals that help control the cell cycle, excuse me, external signals are often called, one type of external signal is called a growth factor, which is a protein that's released by other cells and then stimulate target cells to divide. So again, growth factors activate, of course, a signal transduction pathway. Okay. One example of a growth factor is platelet-derived growth factor, or PDGF. And it stimulates the division of human fibroblasts, which will form skin cells. Pardon me. So if we take, this is just kind of a little experiment to show how PDF works. Take some human connective tissue, chop it up into pieces with the scalpels. So then we have our free fibroblasts in culture. If you put those fibroblasts into cell culture vessels without PDGF, they just kind of hang out. If you add PDGF, platelet-derived growth factor, to some of the vessels, you'll see the cells grow and divide, and you get nice, pretty-looking fibroblasts. So it's secreted by another cell, and it binds to the surface of the cell, signals a signal transduction pathway, and tells the cell to divide. So that's the response. So PDGF is one external signal. Another external signal is density-dependent inhibition, which talks about the density of cells, how if cells get too crowded, if there's too dense of cells in an area, they stop dividing. Another type of external signal is called anchorage dependence. And so cells have to be attached to something, to some kind of a substrate in order to divide. And of course, cancer cells exhibit neither of these processes. They can just do whatever they want whenever they want. And here, if you look, anchorage dependence, so see how the cells are kind of attached to a substrate in order to divide. And then they divide until they kind of fill the bottom of the dish. And once they fill the bottom of the dish, then they stop dividing. And then if you scrape a little bit off with a spatula, they will fill the bottom of the dish again and then stop dividing. So this is, again, what happens in normal cells. With cancer cells, the cells just keep growing and dividing, and they form a just big, huge pile. Okay, and they float around and they divide, and they just keep dividing uncontrollably. And that's why we'll get cancer. Okay, so again, loss of cell cycle controls in cancer cells. And hopefully you're starting to see that there are so many different ways to control the cell cycle that there are, when you get cancer, it could be one of hundreds of things that have gone wrong. Or a few of hundreds of things, or hundreds of things that have gone wrong with the cell that allow it to divide uncontrollably. Okay, but the end result is cancer cells do not respond normally to control mechanisms. Okay, they may not need growth factors at all. They may make their own growth factor. They may give the signal of a growth factor without the presence of a growth factor. They may have an abnormal cell cycle control system that doesn't need growth factors. If you take a normal cell and convert it to a cancerous cell, this process is called transformation. You're transforming it to a different type of cell. Okay. Now, this happens a lot, and normally your immune system takes care of cancer cells. However, sometimes the cancer cells grow too fast, or it cannot, your immune system can't get rid of them. And if they're not eliminated, they begin to form tumors or masses of abnormal cells within normal tissue. If the abnormal cells remain at the original site, the lump is called a benign tumor. These can usually be cut out, taken care of, and you're good. If the tumor is malignant, what that means is it invades surrounding tissues and can metastasize, which means kind of break free, hop in the bloodstream, and go anywhere and form a second tumor in other areas of the body. So here's a little bit. So here we have a tumor. If it just stays right there, it can be benign. You can cut it out. Okay. However, if it begins to divide and invade the neighboring tissue, it becomes malignant. And then if it spreads through the lymph system and blood vessels, so some of the cancer cells will break off and get into your lymph vessels or blood vessels, they can form tumors, they can metastasize and establish new tumors in other parts of the body. So it is a nasty business. Okay. Again, scientists continue to try to understand the cell cycle, and that helps to lead to advances in cancer treatment. So figure out how to reset the cell cycle or reestablish cell cycle control. Okay, and on that happy note, that's it for chapter 12.